only, 1 marks question from 2017 question paper. Okay, so here it says, which one of the following techniques cannot be used to determine the sequence of a novel protein? Okay, so you want to analyze the sequence of the protein, so they are asking which technique cannot be used. So 4 options they have given. Okay, so first option is de novo sequencing by ESI MSMS, mass spectrometry they are saying here. So mass spectrometry, we know that there they purify the protein, they analyze each and every atoms of the protein, and based on that they figure out the uh, sequence of the protein or the residues that are involved, so that gives an indication about the sequence. So this is the de novo sequencing is correct option here for the to determine the sequence of the protein. So this is a correct option. Next is the Edman degradation option. So Edman degradation also we know very ancient technique where you provide a Edman reagent, that is I think phenol isothiocyanate, so that binds to the end terminal of the protein, and that end terminal gets detached, leaving the remaining peptide chain. Okay, so this end terminal and other peptide chain, so this isocyanate, this makes a bond here, and ultimately this leaves in a cleavage here. Now this particular one with the entire back amino acid comes off. Okay, and this particular is now separated and identified like which amino acid it is. So that is done by further Sanger sequencing also. So this also tells you the sequence of the protein. Next, this Sanger sequencing also we perform to find out the sequence of the protein. So protein or DNA, you can say, if DNA is sequencing can be done, then protein sequence can also be predicted from there. So all these three are used for the sequencing of a novel protein can be used. Okay, and the fourth option here is the peptide mass fingerprinting. So here in the mass fingerprinting, peptide mass fingerprinting is uh, done when very large protein is present, very large protein, and using peptide mass fingerprinting, the protein is cleaved into smaller peptide fragments that can be further analyzed by sequencing. But uh, peptide mass fingerprinting is only used for the fragmentation of the protein, not for the sequencing. So out of these 4 options, this particular D option is the wrong option here. So this is the wrong option. Other 3 are used for the sequencing. So this was a very direct question. And this is my NPTEL reference where you can go and read about detailed uh, procedure about the sequencing techniques more properly. Okay. Thank you.